Okay, we'll get started. Good evening, everyone. So I'm excited to discuss about some neural coding problems and some insights from a recent conference. But in today's lecture, we will only discuss this neural coding problem, and on Friday's lecture, we will see into how these things are being used in current research and what all we checked in the new recent conference. So, am I audible?
Yes,
Yes, yes.
Okay. So, we'll just get some like what is the meaning of encoding and decoding here. So, brain is usually in neuroscience or in engineering fields. It is seen as an encoding decoding machine. And what does that mean? So, let's say you sat in front of your piano and you're simultaneously looking at your music notes and then simultaneously humming the song and trying to play on the piano. So here we are doing multiple senses are engaged at once. So for example, like you need to have a tactile sense to get the proprioceptive feedback of which key you are going to play and how your finger positions are. And then your auditory sense is heightened because you need to know what song you're listening to or you're humming, and you want to play along with that. And then now you have to transform this multi-sensory input to like what you want to do next, which key you want to press next, and what is your current state of your fingers, and what is your music, the tone sense, etc. And then convert all this together, which converts into a uh, set of motor skill in action. So, if you have been to play this piano or any musical instrument, it needs all these kinds of senses. You need tactile sense, auditory sense, and your brain has to transform all of this into a goal directed behavior to produce a soothing music or a melody. So, according to this paper, a recent review here, it says that this shows the brain's ability to both encode the stimuli, that is, we need to encode what you're looking into your notes, uh, which color, which keys you're seeing, what is your current environment. Your brain has to encode those sensory stimuli, what tone you're listening to also, and then decode this. Your brain has to, the downstream areas has to decode this into a meaningful representations, so that you can convert them into goal directed actions. And also at the same time, you have to remember the song or notes or what you're playing, and along with memories you get other emotions as well. So it's the involvement of whole brain in this task, and your brain is continuously encoding the stimuli representing your current state and decoding it into a meaningful action. So this example might give a sense of what I mean by encoding and decoding, but it will be more clear when I go into the details. So, for example, in the same task, when you're trying to play a piano, your primary and lower sensory areas. So this, the top picture represents the visual areas, visual processing pathway, and the bottom one represents the auditory pathways. So which is from ears it goes to brainstem, and brainstem to the auditory cortex, here as shown in the cyan colored highlight here. So the first processing areas are generally called as primary areas, either to visual or or the auditory. And now these sensory areas, the primary sensory areas, have to encode the features that you're currently seeing, or the tone you're listening to, or the hum you're reciting. And then the immediate downstream areas try to decode this encoded stimulus. So encode stimulus, how does a region encode stimulus? It is encoded in the activity of certain neurons which are in this areas, and then downstream areas are the ones which are right next to this primary areas, which are connected to right next, and the information flow goes from primary to downstream areas, and then finally to a higher sensory areas. So your higher sensory areas and downstream regions has to decode and build an internal model of your current state, right? And then not just represent or build a model, but they should be useful for your next goal or the immediate goal, to play a proper note or a proper tune or proper set of motor actions. So now this useful representations represented by higher sensory and downstream sensory areas are integrated in this posterior parietal cortex usually. Uh, so and this representation is then downstreamed into prefrontal cortex for movement planning and premotor area for movement planning. So there is some useful representation of multisensory input from different senses going through your prefrontal and primary motor area, premotor area, and then your primary motor cortex, which sends the command to the through spine to your muscles to play the tune that you're currently trying to play. So this entire thing encodes different stages of encoding, decoding, encoding, decoding. So first encoding is just the sensory input to represent your state, and this is further decoded to useful representations for your motor areas to perform goal directed actions. Okay, in the author's words, collectively this process of neural coding and encoding and decoding lies at the heart of fundamental neuroscience, and this gives a primary framework for how brain computes to perceive, act, and learn. So this is the main framework, and by this framework, I hope everyone is maybe somewhat clear with what I meant by encoding and decoding. Right? Any questions here until now, or any comments which you want to share about what you think about this framework? Okay. So we'll get into the details of these problems. So these are called neural coding problems because your neurons, basically your yes, every area. This okay. So Ishita is asking, even higher sensory areas can encode, right? Yes. So yes, for this particular task, I meant the encoding and decoding stages are this way. But what I mean by encoding and decoding becomes more clear. Then you will know that every area can encode and decode also. But rather than focusing on how the brain processes, we are trying to make use of the representations in the brain. So whatever I'm going to discuss is little far from this, how I presented this. So you can call encoding and decoding simultaneously as well. So when I say area encodes, it can also decode. So but whatever we are going to talk is more of correlational than causational. But whatever I presented right now is a causal framework, like brain encodes, and because it decodes, you are able to perform action. But whatever we are going to discuss, the techniques that we are going to use for neuroscience are more of correlational, but they have a potential to explain this causal frame. Okay. So this is one neural coding problem is encoding, as we saw here. So what happens is your neural activity can be recorded through multiple paradigms. Either you can record your neurons membrane potential or the spiking activity, or you can do electrocorticography or fMRI or calcium imaging, anything. So your brain generates some activity and we are going to record that in any of the paradigms. So that we represent here as neural activity, and we label it as Y. Okay, and your neurons are potentially responding to some stimuli in the real world. It can be your visual input, or where you want to you know uh, where you want to move your hand or grab something, or anything, any sensory stimulus or auditory stimulus, that a tone you are listening to, etc. We'll label it as X here. So now we are going to see, given a stimulus X, what could be the neural activity, or this is encoding is more of a what is a mapping between behavior or sensory stimulus to the neural activity. Okay, so this is the encoding model. So it basically gives the mapping between stimuli or behavior correlates to the activity that you're recording. Okay, yeah, it need not be direct electrical activity, it can be any inferred activity based on the recording paradigm you are using. Okay, the other neural coding problem is your brain generates some signals that you're recording using different modalities. Now, given the neural activity, can I predict what behavior the animal is going to perform? Or so can I simultaneously predict, given the neural activity or past neural activity, can I predict what could be the current action, or what could be the stimuli feature that the activity you recorded represents? So this is called the decoding model. So this we represented by this is represented probability of X, that is the behavior correlate given the neural activity. Whereas in the previous case we saw probability of neural activity given the stimulus. So here we are trying to find the mapping. Here we are trying to, given the neural signature, can you predict the behavior? So these are the two main neural coding problems. There is one more which we'll discuss later, but for now we'll focus on this encoding and decoding problems. So and keep in mind that this is only the referred activity. Whatever we are measuring is only the referred activity or inferred activity. It's not exactly what brain is doing or your neurons are doing, right? So this will always be correlational. So we should not interpret more like a causal role. Because of this activity, you are able to like your hand position is going there or something of that sort. So what are the utility of encoding? Now we'll focus on encoding models. So because encoding models give a mapping between a stimuli or behavior correlate with the neural activity, right? So we have a mapping now. So how is this mapping useful? Any ideas of how can we use these encoding models, or if you are informed of this mapping between stimuli and neural activity, how can you use them? Okay, the answer is here itself. So we can use it to write into the brain. So why do we want to write into the brain? For example, we are all very familiar with this cochlear implants. So what happens is, this implant, this device, has an electrode inside, which first extracts the spectral features and writes to the this implant, and then the neurons connecting to this implant will be the downstream. So right now this implant is encoding the auditory activity, and the downstream neurons which will be connected to this or which are joined to this implant will be the downstream regions, which will decode and then further process by the other brain areas, and hopefully there is some kind of decoding going on here from the representation. For this we need to know what is a mapping between the primary neurons here from the cochlea. Okay, so once you know that, basically here we'll talk about a matrix called design matrix, which is useful in this encoding models. So in this particular case, the design matrix is nothing but the Fourier transform matrix. So you have a matrix with different set of oscillations. So that forms the design matrix here. It will be more clear as we go forward, but this is where one kind of application where we can use encoding models, and this encoding model is very easy because a priori we know what auditory features are important to the brain, that is frequency. But for few we need to build the design matrix. Also recent innovations in bionic eye, that is people who are blind, we can give them a second sight by knowing the mapping between the visual stimulus and how the retinal ganglion cells respond. So basically, okay, we don't give them a full vision, but it's a kind of blurred, or at least the participant will be aware of what obstacles he is looking at. Okay, so we can build, based on this responses mapping, we can build a encoding model which transmits into a device which is connected to your retinal geniculate nucleus, okay, retinal ganglion cells. And if you know the mapping between stimulus and retinal ganglion cells, we can use this for this kind of application. So just for motivation, we'll see a video from Second Sight, a company named Second Sight, where they build this kind of devices, the retinal brain computer interfaces. So let me know if the audio is like you're able to listen.
Sorry, what can you see right now? Nothing now. Nothing now.
Is it audible? Okay.
Everybody ready? Okay. So 3, 2, 1, start stimulation. Yes.
Can you see?
Oh my goodness. Wow.
Can you see, Larry?
Yes.
Oh my goodness. Can I give him a kiss?
Yeah.
Oh, yes.
Wow.
Can you really see?
Yes.
Oh
It's
my
flashing.
goodness.
Big time flashing.
They're flashing? Oh my goodness. That is so cool.
Larry Hester, 66, has been blind for half his life from a condition called retinitis pigmentosa. In September, an electronic stimulator was surgically implanted in his left eye. On October 1st, Duke eye surgeon Dr. Paul Hahn turned it on for the first time.
So the journey is just beginning. This is day one of hopefully the rest of his life. I think turning this device on allows him to experience a whole new world. And it's not vision as we traditionally know it, but it's a whole new type of vision for him.
Are they Oakleys? Is that what they are?
They're Oakleys.
They are cool. Only the coolest for Pop.
That's right.
It was incredible. It was bright, and I just had to take a deep articulate what I was feeling, but I wanted to share it with everybody I could grab at the time and hug.
Thank you for this.
Almost a pixelated type of vision where they are getting stimulated with flashes of light when they perceive something that's bright, and when their camera is picking up something that is darker, then they get less flashes of light.
Yeah. Oh, look, he did it. He went right to it. Woohoo! All right.
That's pretty cool,
What a
huh?
guy.
Amazing.
Yeah. So whatever you saw in this video is there is a spectacle which has a camera, video camera, where it sends the current visual world as an input stream, and there is a video processing unit which actually only takes the features which are very important for just to detect the edges or contrasts in the environment. So once it the video processing unit is reducing the dimensionality of the visual world, so that it can match the number of electrons that are used to stimulate the retina. So the retina has 60 electrodes, so it has to reduce the information using certain bottleneck. Therefore, there is a video processing unit. Then wirelessly it sends that the video processing video processor's output wirelessly to a stimulator, which is used to stimulate the electrodes attached implanted to the retina of the subject. It is implanted on the epiretinal layer, so then it can stimulate the retinal ganglion cells, which is the first cell that receive your visual light has the input, and then through optic nerve it then sends the visual stimuli. So basically the subject is able to see a so this stimulation creates light blobs in his vision. So basically he can see some phosphenes, and the phosphenes have a corresponding one-to-one correspondence between the location where he is seeing in the real world versus the electrode where the stimulation happens. So this gives him a sense, not exactly vision, but a sense of pixelated vision, where he can still see and move around. So this is one of the application for knowing the encoding models, because we will know the mapping between, for a given stimulus or given a world with given contrast in the visual world, how does your retinal ganglion cells encode. So any comments or anything here if you didn't understand some part here? Okay, cool. So basically what we are currently dealing with is encoding models. So we'll get into a slight mathematical formulation of it. So in this lecture we'll be mostly dealing with motivation and mathematical formulations, but in the coming weeks, after next week, we will discuss how to implement using some neural data or EEG data, etc. So basically encoding model here, we represent it this way. It reads like probability of spikes or EEG recording or any neural recording given the stimuli or some behavioral variables. It can be movement, auditory stimulus, etc. And you have a model which does this, and that model has some parameters called which are represented by theta. So basically what you're doing is you're replacing your brain by a model which is represented by theta here, which are the parameters of that model. And now you give a stimuli and it will output the spikes or neural recording as an output. So now what we have to do is we have to learn the parameters theta. So basically we have to learn the model to know what is the mapping between stimuli or task variables versus the spike potentials or the EEG recording or MRI signal, okay, fMRI signal. So just let's take one example and use one kind of a distribution which is mostly used in neuroscience, because neurons have that characteristics which are described by Poisson distribution. How many of you are aware of Poisson distribution? Okay, cool. So this distribution is represented this way. Let's say probability of k, some variable, given a parameter lambda, is equal to, this is a mathematical notation, but if you plot this for different lambda values, you get this distribution. For lambda equal to 1, you get a exponential kind of distribution like this. Then as you keep increasing lambda, it becomes more of a Gaussian kind of thing. This is a Poisson distribution. Where is this useful? So Poisson distribution is useful where in a certain window there is a constant rate of for example, we can take an example of call drop. So let's say a telecom operator observes on an average true drop calls per minute. So in a 1 minute window, average rate is constant. So in this case, when the average rate is constant and the probability of drop is very low, in those cases we use a Poisson distribution. Same thing goes with a neuron. So a given neuron, we assume that it has a constant rate of firing in a given window, average, on an average. But the probability of firing in that small window itself is very low. So in those cases, we use this Poisson distribution, and because neurons characteristics are like that, we use this Poisson distribution mostly. So for example, if I tell that this call drop follows a probability distribution, if I ask what is the probability of observing 3 drops in a minute, so for this in this formula you plug in 3 and see what happens. Okay, and the lambda value is nothing but the average drop rate here. So when you do this, you can get the probability of. Okay, so here basically x is the k here, so don't get confused. Okay, so is everyone clear of what and when we use a Poisson distribution? Okay, cool. So now we can use an encoding model called Poisson linear neuron model to encode the spike counts for a given stimulus parameter. So let's say that stimulus parameter is the contrast. Okay, so Poisson distribution looks like this based on a given mean and variance, which is the lambda parameter here. Lambda is both mean and variance for the Poisson distribution. So a spike count can be represented by the Poisson distribution with a average spike count parameter lambda, and we have a stimulus, and the model is linear here, so it's just a multiplication of the model's parameter theta with the stimuli. Let's say stimuli is the contrast here, different levels of contrast. So we do a linear transformation, and it is represented in the parameter lambda, and this lambda is used in this Poisson distribution so basically we are encoding in the mean firing rate. The stimulus via model is encoded in the average firing rates, and this firing rates we pass it to a Poisson distribution to replicate how the neuron respond to the given contrast. So here the full encoding model for a given neuron is represented by this when you plug in the x and y and theta in these equations. So lambda is equal to theta x, and your y is the the spike rate, and x is the stimulus contrast, and theta is the parameter. So here given if you collect some data by showing some visual contrast and then collecting this neural recordings, you will have x and y, but the unknown here is theta. That is the model. We need to find theta, which maps from the stimulus to the spike count or the neural recording. It can be anything. It need not be spike count. It can be EEG signal or fMRI responses. So how can we visualize this conditional distribution? So we can visualize this conditional distributions in a two-dimensional plane as shown here. So here x is the stimulus contrast. Given x, you want to know what is y. So we can represent a Poisson distribution for different levels of contrast this way. So we don't know yet how the distribution looks like. This is just to know how can we visualize. Okay. So so when you collect data, you will get some points like this. So for different contrast, you want to see how the response is. So you can get some data points. You can't get the whole distribution, but you get some data points while recording. And this is just to show how we can represent this conditional distribution P of y given x and theta. So okay, this is a distribution, and you might, if you record, you might get these points like I showed here, some data points. You can't get the whole distribution. You get some samples from the distribution. Okay, so now we will get directly into the maximum likelihood formulation. So this is one method, maximum likelihood estimation we use to find the mapping between stimuli and the neural recordings. Okay, let's say we do some experiments. we get all the spike counts for a given stimulus. For a given stimuli, you in different trials you present different stimuli and you get all the spike counts, and then the model has some parameter theta which you want to find. So you want to find theta which maximizes this probability distribution. Okay. So now previously we saw only for one neuron, but sorry, it can for one trial, but it can be for many trials. So what we assume is every trial is independent from each other. Therefore, the probability distributions becomes a product. Okay. So this whole probability distribution can be represented by this equation. Pi represents the product of each condition for every trial. Okay. For every trial you get some data, you just do the product of it. Okay. Is everyone clear with this formulation? Great. Okay, so if you don't understand, you can please ask. Even in next slide, if you get any doubt, you can ask. Okay. So we are just expanding this to multiple trials. Whatever we discussed here, we are expanding to multiple trials. Okay. So now, let's say you have some data like this red point. We focus on red points. So we have some data red points. Now we have to fit a model to this. So what parameter of theta best fits the recorded data? So for different contrasts, so let's say you have shown 3 contrasts in the experiments. So for different contrasts you have collected this data. Now what you have to do is, the intuition is, you have to change the parameter theta such a way that this distribution aligns with the data you have. So that is what the maximum likelihood estimation is. Okay, so basically we want to fit a distribution to the given data or the observed data. So for this I can, let's say I have, hypothetically, let's say I have a knob which I can turn to rotate this light the white path here, which is the visualization of conditional distribution. Okay, for theta is equal to 1.5, I get this kind of a 2-dimensional plot. Okay, I can turn a knob differently. Yes, so in multiple trials you show multiple contrasts. So this is repetition of the same contrast multiple times, or these are at different trials you are showing different contrasts. So yes, so each point depicts a trial here. So this is just a visualization. This is not exact data, but yeah. Okay. So let's say you have a control of this knob where you can control where this white patch goes. Okay. So initially you put 1.5, which is little off from the data you collected. Then you put 0.5, which is going below the data points. You keep on changing until you find an optimal theta which exactly explains the data that you observe. So in this case, when I put theta, when I turn my knob, theta knob to 1, I get visually here we get a very aligned 2-dimensional distribution to the data that you observe. So basically this is the maximum likelihood intuition. So we will get into how we get there, but is everyone clear with this intuition of what we are doing and what is meant by trying to find the optimal parameter that fits the distribution of the observed data? Okay, cool. So this basically we have made many assumptions that trials are independent, neuron is a Poisson neuron. So because of these assumptions, we can simplify the problem and get a closed form solution when you apply some mathematical formulation. So the likelihood function that we saw is P of y given x, P of all the neural data given all the stimuli for a parameter theta, you get this function. And now you have x and y because you have performed an experiment. Therefore this function is a function of theta. So it's not you can represent it as probability distribution as this, or you can write it as a function of theta, let's say L of theta or something. So you can keep changing this, either probability way of showing it or likelihood way of showing it. and because we saw across every trial is independent of each other, we get a product of each. Okay, and when you expand it, you get this kind of a equation here. Okay, in general, when you have a product of multiple things, it is usually complex. So what we do there is we take a logarithm. So what if you're not aware of logarithm, just remember here that the logarithm converts products to sum. So when you take a logarithm of all these products, you can write it as sum of all each of this inside variable, and the power goes into the multiplication factor here. So theta power yi will go into the multiplication factor in front here. So basically it gives a concise equation for further evaluation, so that you get a closed form solution easily. So just remember, from likelihood we are going to log likelihood. This is usually what in general we do in any, not just in neuroscience, but any of the maximum likelihood estimation problems. Once you get this, so now we have a function of theta. We want to find theta which maximizes this whole function, which gives a maximum value, so that you're finding the parameter theta such that it exactly matches the mapping between x and y that you observe in your data. So can anyone give an idea of how to find the theta which can maximize this function? Any idea from your 11th, 12th grade calculus or something? We have a function of theta. We want to maximize it. So any idea of how to maximize a function? Okay, fine. So is everyone clear until this point how we got? Yes, correct. So as Indraja mentioned, differentiate and find the root. So basically we have to differentiate this function with respect to theta and try to equate to 0 and find what value of theta maximizes it. So once you simplify this using some basic mathematical identities, you get some form of log likelihood function of this sort. Then you differentiate with respect to theta and equate to 0. Then you find the maximum likelihood estimate of theta is nothing but sum of all the firing rates that you see divided by sum of all the contrasts that you see. So basically this reduces to a weighted average kind of value, weighted average of each neural response with respect to the reciprocal of the contrast values that you see, sum of contrast values. So no, I think it's just it's just this one. Don't overinterpret it. So we got here an expression for theta that will be the maximum likelihood estimate of the model. So this theta is a mapping between the neural responses versus the stimulus when you use a linear Poisson model. So now this was an assumption that we are using, that it's Poisson or using a Poisson distribution. So there is no reason to not use any other distribution. We can use Gaussian linear model also. So these are all approximations of approximate model of how your brain encodes the stimulus, but it's still not causal. So for a Gaussian equation like this, the mean firing rate is same as theta x, some linear transformation of the stimulus as you see here, and you have the data of firing rates and you have this distribution. Now here we have to similar to the previous problem that we saw, we have to find the optimal theta. So similar way we try to build the log likelihood function. When you simplify this, take log and do the product, you get this kind of a equation. And when you try to maximize it, you get a maximum likelihood estimate of this sort. Okay, so it is similar, but you have a product term of both the what is the stimuli and the neural responses by the squared of the stimuli responses, squared of the stimuli parameter. For Poisson, we saw it is weighted average like this, but for maximum likelihood estimation, what is the intuition behind this equation is we are trying to correlate the stimuli with the neural response. Okay, it's this is like a dot product. So if they are maximally correlated, you get a high value. If they are not correlated, you get a very low value. So based on that, this has a term also called correlative learning. So you try, it's like, or coincidence detectors. So you try to coincide two things which might potentially might be related, and if they are related, you get a maximum value, and sum across all, take an average across them, normalized average. So this is the maximum likelihood estimate for this. So you will later see that this is also a solution when you represent it as a matrix, the least square solution. So we'll get into it, no need to worry about it now. But yeah, is everyone clear until this point of what is maximum likelihood? If don't worry about the equations, just if you get the intuition of this maximum likelihood and what is meant by it and what are you maximizing. Okay, cool. So now we have assume few variables. You might have a question that how do you compare this? Which one is the best? So there are many methods to validate a model. We'll not discuss that right now. When we are dealing with the code, we can discuss how to evaluate. But there are multiple ways to evaluate, but everyone can have a question that which model is the best, or which mapping describes the stimulus to the neural response best. What is the best mapping? So you need to do some evaluation methods to evaluate models and get the best out of it. But what if it doesn't follow a particular, it's more general, it doesn't follow any, you don't see any pattern of a distribution here. So let's say your data, the data you collected have a profile like this. Okay, for different contrasts, you have the responses you have collected is this way. These are the data points from your experiments. Okay, so now what we do is, so this clearly looks like a non-linear mapping. So there is no a linear increase or decrease kind of thing with the contrast. So one way to model this non-linearity is, instead of just multiplying theta x as the average firing rate, you pass it through a non-linear function, some function which is non-linear, okay, some f of theta x. So this says firing rate is non-linear, and then similar to the previous fashion how we saw, we'll just pass it to a Poisson variable here, okay, and you get the spike count here. Similar fashion, everything is same except that this is a non-linear function. Instead of just multiplying theta into x, you're taking a function of linear mapping of theta into the stimulus parameter x. That is the contrast. X is a contrast. Okay. So basically we have, right now we have discussed, then you can, once you get this formulation and you have an assumption of what f could be, let's say it looks more of exponential, you can put an exponential curve here and try to do the same similar things that we do, compute the log likelihood, maximize the log likelihood, and find the parameter theta. So now we have seen two kinds of models. One is called general linear model, where the stimulus parameter, the contrast, is just multiplied with a theta. So basically we are doing a dimensional reduction here. It's also called a dimensional reduction, but this can be called by many names depending on what you're doing. So we multiply theta with x, and there could be some noise always, so we add some noise and we take this as y. So this noise, if you assume it to be Gaussian, you'll get a Gaussian distribution. If you assume it to be a Poisson neuron, you get you pass it through a Poisson variable and you get whatever expressions we discussed in the beginning. And just now what we saw is a generalized linear model. That is, if the neuron is like it is very hard to assume a particular distribution for the responses you collected, and there could be some non-linearities. Then we use a generalized linear model. So there is a stark difference between this, that there is a non-linear function coming into place, and then you follow the same procedure of finding the theta, the model's parameter. So these are the two formulations. On this, once you build the generalized linear model, you try to find the parameter and try to maximize the likelihood function based on the data that you got from your experiments. So this is a formulation of maximum likelihood with generalized linear model. So now we have learnt about the concept of maximum likelihood estimation, finding the best parameters for a model, and what is the general versus generalized linear model. Okay. Is everyone clear until this point? Any doubts, any questions, or if nothing went through, please ask. Okay. I see only 2 thumbs up. Hopefully others understood. Okay. So now we can get into more, like we were using a simple parameter called contrast here, but we can use a whole image as well. So let's say x of t is some image, and we can use a stimulus filter assuming that only certain features of an image is important. So let's say you have a some image of this size, and only these parts of the image are important for that neuron or for that population of neuron. Okay. So what we do is we just pass it through this filter, and only important things come out, which are important for the response. And then you have some nonlinearity. We generally here we do exponential nonlinearity, and then pass it through the Poisson distribution to get the spike counts or spiking responses here. So this is a summary of a LNP cascade model, which is called linear nonlinear Poisson model. So which is generally used to model the neuron's responses, a single neuron's response, given the stimuli parameter or image or auditory parameter, etc. So one assumption is the spiking distribution is always Poisson. Therefore, we use a Poisson distribution here. Okay, we can use any distribution here based on how the noise structure is. So noise structure in the case of neuron spiking is Poisson. So that is how you get. So even if I present same stimulus multiple times, the spike responses need not be same, because it's a probability distribution. So it's not always exact. It always varies. There is always a noise in the picture due to various factors, inherent factors, external factors, etc. Okay. So let's see little more in higher dimension in action how this looks like. So let's say some stimulus parameter is there, which is represented by this blue time series. Let's say contrast only. So at different times I show different contrasts. Okay, or different levels of maybe some tactile force which is being applied to proprioceptors of a animal or a human, and those levels are represented by this as the time progresses. And then from a neuron you're recording, and you have the spike trains. So spike occur at certain instances here as shown here. Okay. So now what you do is, let's say this is my window of interest, and I want to know what is happening given the previous window of this stimulus. Okay. Then I keep shifting this window, this red colored dotted line, I keep shifting. Okay. I keep shifting this by time lag increments of 1. So I shift, I get in the next time sample I get a spike for this kind of a previous previous stimuli waveform. and so on and so forth. I can shift time window by 1 sample until like 5 samples here. And so if you take each of this window, you can construct a matrix. So that will be our design matrix. So it will be more clear right now. So what we have on the y variable is the spike in each window, each time lag. At each sample you have either you have spike or you don't have a spike, which is represented by this vector here, 0, 1 vector. And then based on what window of stimuli waveform you're using, you get a matrix like this. So now you have the data, the stimulus and corresponding responses, and you have to find some parameter k. There is also some noise such that the values in the k maximizes this mapping. This gives best mapping. It can give a best mapping. So usually the variables in k are called regressors. So this is also what Atharva was dealing with when he was teaching linear regression. So we have to find the best. Can the window of response be this? Ishita, are you asking if the window can be of same length, right? Yeah. So so there is, it's like a hyperparameter, the time window that you select. There is no reason to select this particular time window and only this particular time point. You can select whole window, but right now, just for explanation purposes, to keep it simple, I'm using one single sample as the spike window, and the whole the length that you see here as this. So based on how you construct this problem, you can define the design matrices and response. So in some cases, the response need not be a vector, it can be a matrix. So in the case where you're telling the responses will be like a matrix, where you can, wherever spike occurs, you can put an identity matrix, and then you can match the windows of both, or you can take whole row as time and repeat it multiple times of that sort. Okay, we can discuss this offline, but to keep things simple, I'm just using this. Okay, so what's the time? Okay, so basically when you do a maximum likelihood estimate on this kind of a formulation, as I mentioned, it's a least squares estimate. So you get when you do the mathematics, you get to a solution of this sort. Okay, so this defines, x, y, and when you compute this expression, you get the optimal k, and this is also a maximum likelihood estimate, or it is also equal to the least squares estimate. That, I think, if you're aware of Atharva's lectures previously, there were in the linear regression model, you will find this as the orthogonal solution, not orthogonal solution, the optimal solution for a least squares problem of this sort. So okay, now I will just ask a question here. Let's say x transpose x here represents the stimulus covariance, and let's say they're all independent and there is no covariance between them. So this turns out to be an identity matrix, the x transpose x. Now if I take x transpose y, can anyone tell what it represents? So I'm just multiplying transpose of this with the spikes that you get. So I'm just multiplying transpose of this with this y. So can anyone just tell what intuitively it means? Like, just if I do x transpose y. The hint is also here, called spike triggered average. So the hint is in the last word of the term here. Any ideas or any approaches which we can use if this is an identity matrix? let's say this is gone, this term is not there, only x transpose y is there. Uh, anyone wants to speak up or type in the chat? Okay, so basically it's the name says spike triggered average. So what we are doing is wherever there is one, that particular design matrix is you take an average of it. Hello, am I audible? Okay. So basically what we are doing is wherever there is one, in the corresponding window we get an average of it. So we can then say that when you average all those windows where the spike occurred, you see you get an average waveform of this sort in the same window that you are using. So basically then you can say that this particular neuron responds to a stimulus waveform of this particular sort, or this can also be extended to some noise. Let's say I am looking at visually responsive neurons, and I show some white noise screen, and each white noise screen has some particular pattern of white and black patches. So now if I do a spike triggered average, I will know which particular sum of average of particular noise patterns. I get one particular stimulus. Let's say on a whole dark scheme I have one blob. So then I can map for which area in this stimulus field the neuron responds to. I can get through this spike triggered average. So I don't know if that is clear. It went a little bit in the air, but yeah. So basically I want to say that if you average all corresponding one corresponding blue waveforms here, you get one waveform. That average waveform is what you can tell that the neuron responds to. Okay, or that particular stimulus is one that neuron prefers to respond. Okay, that is another intuition behind this spike triggered average. Okay, to know what does a neuron respond to, or a population of neurons respond. Now also you can like as Ishita pointed out, how can we increase the window, or can we increase the window of spike neural recordings itself. So in that case your model becomes even more complex, where you put a feedback of delay line. It's like delay line, like history also going as an input to this model. So you can make this model as complex as possible. So we saw one neuron, we can extend it to multiple neurons with different receptive fields coupled via some filter parameter of this sort, and you get more complex and complex response, and you can make it as complex as possible. But usually simple models can explain very well. So this is a depiction of how a complex model with multiple neurons and histories look like. So this is the direct stimulus parameter, this particular region, and then you take neuron 1's history, neuron 2's history, and so on, and you stack it. So this is how the design matrix looks like when you have a complex model of this sort. So instead of waveforms, we are presenting this contrast as a black and or grayscale stripes like this, and then you have a spike responses. You need to find the regresses which will map between the design matrix and the spike response. So the key to take here is the design matrix is very important, and how you construct that changes a lot between the mapping between the stimuli and the neural responses, so than any other parameter. Or so we have discussed when it's a closed form, the log likelihood, it need not be closed form. In that case we do a algorithm called gradient descent, which we will deal later, but it's like step by step going to the minimum. You can do multiple iterations and go to the maxima of the parameter, which can maximize the likelihood function. Okay, so as I mentioned, we can use this for using, okay, another application of this is estimating the receptive field. So what is a receptive field? Let's say for every neuron there is a receptive field. That is the visual field in which the neuron responds most to. So at this blue regions its response very at the baseline, and at the red regions the neuron responds very high for a given stimulus. Okay, so we can use this same formulation of maximum likelihood estimation to find the receptive field, because this is the parameter theta that we are trying to estimate. Okay, so this is the theta is a receptive field parameter, and we are using an exponential linear Poisson model, and once you do a maximum likelihood estimate you get the theta, and you can know what the receptive field of a neuron is. So is everyone with me until this point, or at least if they understood partially is also fine, or if you have any doubts, comments, please mention. Okay, cool. So we can use this general linear models or generalized linear models for EEG as well. So right now I will not go in through this, because we will have a notebook upcoming for this, because like our focus is also on EEG rather than spikes or fMRI. So what we can do here is, this is a design matrix here. So you let's say you have two stimuli conditions or two stimuli types. So stimuli type 1 is this way. This is a design matrix for this, and you have, for a given electrode you have this kind of responses from different trials, and different trials also have a different noise levels, and there can also be some parameter which is continuous, or some or you can have a constant value to do the offset of the EEG responses. So this is another linear model which you can use it to get like what does the ERP of a given condition or given stimulus type look like. So instead of always locking time locking everything, what we can do is we can do this kind of a general linear model to estimate the event related potentials as well, or how the noise for a given noise parameter noise levels, how does the ERP look like. Or you can do multiple things, but this is one kind where you can extend this to not only spikes but also EEG or any neural recordings. Okay, this might not be very clear, but yeah. So now with the advent of many algorithms, machine learning, deep learning, they have many advances in doing this encoding models as well. So this is one paper where so what they did is, the participants are presented with multiple natural stimuli like this, natural or artificial cartoons, etc. And the participants fixate here, and he continuously sees many objects, and we collect the EEG data. Okay. So what we do is, on one stream here, the EEG data is collected. Okay, and you get at different conditions. Like here the conditions are different types of stimuli, like animate objects, inanimate objects, animals, birds, etc., reptiles, etc. So different conditions or trials, time, and different electrodes. Let's say if I'm using 17 channels, 17 electrodes. So I have the whole three-dimensional EEG data that we initially saw in the very first lectures. Okay. And now on the other hand, there is a deep neural network. Okay, some neural network or some weights, let's say some parameters here, which maps the stimuli to some activation as shown here. Okay, some output, it gives some output. Now you try to do a linear mapping and try to minimize the error between the EEG time series that you got versus for every time point and channel. Okay, you try to minimize the error or the difference between the actual EEG and the synthetic EEG from the deep neural network, so that you learn the weights, let's say. And now in the test conditions, you have some test images, and you pass it through the deep network, you apply the same learned weights to get some vector like this. Now this vector you can construct for every time point in channel, and you can get a synthetic EEG. So now what we are essentially doing here is we can replace our brain with a model of neural network, and we can know how can or predict how excuse me, how can the EEG response look for a given test image. So this is one application where we can use the encoding models to know the mapping between the stimuli and EEG responses. This can also be used to augment the data. Like if you have very less EEG data, but you want to do very good predictions and you don't have good amount of data, you can use this encoding models. You have some responses, actual brain responses, actual EEG responses, generate some synthetic responses, augment it, and then train even complex models. So this is another applications, another little high level application of these encoding models. So is everyone clear with this, or any ideas or any doubts here? Okay, I think this went a little bit in air, but so basically here we are trying to get the mapping between the stimuli and the EEG activity, and with that mapping we are trying to generate a synthetic EEG data. Okay, or this can also be a prediction, like you can predict how an EEG response looks like for a given test stimuli. Okay, instead of using linear weights or non-linear functions, we are using a neural network here, deep neural network. You can think of it's like multiple transformations with the weights. So multiple non-linear transformations based on the number of layers the network has. That's it. Deep neural network is nothing but a non-linear function with applied multiple times in sequence. Okay. So there are different ways to optimize and learn it, but right now we are not going into it. So another famous research article is about like from large scale neural population recordings can we predict behavior. So this comes more of a decoding, but also you can predict neural activity that way. So let's say you have a behavior for a given behavior, some parameter you extract. So what we have is mouse movements, video. From video you extract some features which are shown as PC1, PC2, PC3. Basically on video data we are doing a technique called principal component analysis, and you get this. For each PC you can know what a neural response looks like. Okay, or you can learn a model like a regression model and try to find for a given video feature how does the response looks like. So this is not even seen, this is just a mouse behavior and its default spontaneous firing rates. So here they showed that with just the behavior, some feature extracted from the behavior, they were able to predict the neuronal activity as well. So which is close to the recordings that they perform. So from this eigenfaces, they got a prediction of neural activity, which is same as the recordings during the similar behaviors, how a neural activity looks like. So this is just an example of research to show how encoding models are used. So once you have this, it can act like a model to generate more and more synthetic data, or to form new hypotheses of how can a neuron respond to without doing any costly experiments, or costly validations for for this bionic eyes or cochlear implants, etc. Okay, we can just use this learned model to learn the mapping between stimuli and this. So now coming to a bit of philosophical part of this, like whatever we saw is only a descriptive statistical model. So it doesn't have any causal. It should not have any causal interpretations. Like we should not over-interpret them that uh, this particular behavior is causing this activity, or this particular feature is causing this activity of the or the response. It's just describing the mapping. Like this feature, the neurons respond this way. Okay, it doesn't say as an why the spike have to be this form, or in the first place why we assumed a Poisson distribution. So those all are more higher level questions, which comes through uh, like Poisson distribution is efficient way of coding information, so that with very less amount of energy you can represent any stimulus. So that is why neuron is Poisson. But why is that? The concept itself is a little far away from what we are discussing right now. Or how is it implemented? Like how the neurons are related, how excitatory inhibitory coordination is there to generate this particular spikes. So those are next level, but this describes the three levels of neuroscience. So mostly computational neuroscience deals with describing the mappings between stimuli and responses. Then if you go further deep, they can find the mechanisms which say why the firing rate has to be this form. Or if you go to connectionistic approach, they will go more into how it is implemented. So this is also in line with David Marr's three levels of approach or analysis. So one is computational, why do things work? Or this comes like if you can build a model which can exactly replicate it, then we'll know why is this way. Right now we are dealing at only algorithmic level, where we know the mapping between stimuli and neural responses. Then other stage comes at the implementation level. So can we implement all this into a hardware and make an artificial brain out of it? So those kinds of stuff. So is everyone clear why this is still not causal? So to put in other words, just because you can decode a stimulus doesn't mean that your brain is also seeing that way, or your downstream regions are seeing that way. So yeah, there are people who say like brain does some kind of clustering or some algorithmic implementation, but it need not be true. It's still an hypothesis, and our models are not exactly the brains. So this is just explaining, like it's called principle of redundancy. So with multiple models you can get the same output. So it is that way, and brain is one part of it. So it's still not causal, and the motivation that I presented in the first seems to be more of a causal, like brain is decoding kind of thing. Brain is decoding, but in a different way, not like maximum likelihood estimation or anything of this sort. Need not be anything of this sort. Okay. Is everyone fine with this kind of, like, if you have any opinions about this philosophy or anything? If you agree, how many of you agree with this? Okay, Abhimanyu, you can unmute yourself.
Yeah, I'm outside actually. I can't. It may not be clear.
Oh, I think it's fine. You can speak. If it is not clear, you can type in the chat. Okay. So, meanwhile, we'll just see other kind of models. So now we are going to decoding model. So what is it? Given a neural activity, can you predict the behavior? So this comes to the decoding models. So what is the utility of this decoding models? So it is useful to make brain-computer interfaces or processes. Previously we saw writing. This is like a reading of the brain, reading of the neural responses. So this is a recent advancement in speech neural processes. So what they did is they have a neural network which learns the phenomes encoding from the neural responses. So it takes in the neural responses and gives out the phenome or phonetics. There is another neural network taking the same neural recording, giving the speech sound features, like how does the speech sound when you speak. Basically the audio waveforms or audio spectrograms. And then same decoder, with the same data, there is another deep learning model which can map to the muzzle movements, the tongue, the jaw movements, etc. when you speak. So these neural responses are taken in and can synthesize speech, can synthesize the text on the screen of what you're trying to speak, and it can also make the movements, the jaw movements on an avatar. So this is a recent research where a patient with brain stroke who cannot speak was implanted with this processes, and they trained these models on different data sets on her data. So as a child she was able to speak, so they have those video recordings which was trained from, like, the movements from there are taken to train this model. She has the voice when she was a child before she met with some accident, and so based on that data from her previous data, previous videos, etc., it was trained, and they also augmented with other data set available. And this is a high gamma activity. So here they show which responds to what kind of jaw movement, which electrodes give highest activity to the high gamma activity. So this motor movements are known to show high gamma activity. So based on this, the model can decode and find how to move the avatar's muscles while the participant is trying to speak. Let's see a video of this and then we'll stop, and remaining parts we'll discuss in the next lecture. Yeah, so we'll start the video. Yeah, you can read as well.
I think you are wonderful. Give me a few minutes. Do you think of my artificial voice?
So basically here in this video the screen is prompting what for the subject what to speak. So and then she can't speak, but she tries to prepare for the speech, and as she prepares for the speech, the array implanted in the brain records the electrocorticography, which is on the cortex close to the speech area, tries to see the activity and decodes the speech, and in feedback gives to the avatar, and avatar tries to speak instead of the participants who can't speak or the patient who can't speak. So this is an example of decoding from neural responses. So you have ECOG responses, electrocorticography, from the cortex, which is close to the speech area, which is put on the surface of the cortex, and it has some 253 channels, and based on the activation patterns we are trying to decode the speech. And there are very complex models being used. Right now we are dealing with the simple models, but this simple models becomes the basis for building more complex models. So basically, if you take regression in this case, you have to reverse the terms. Instead of x being the stimuli, here y will be the stimuli, and x will be the neuronal data or brain data that you record, and you have to find the optimal parameters. So if you're doing regression in this case, you have to reverse, and the same formulation applies. The same maximum likelihood formulation applies here as well. So this is one technique to decode. We'll deal with others. So if you have any comments, or if you feel something is exciting, please you can please share, and then I think we'll end the lecture here. Or any comments, any doubts? Okay. Abhimanyu, you have your doubt type or something, or you want to ask later?
Yeah, I think I'll ask later. Yeah.
Okay, fine. So that's it for today. We will deal with some different frameworks for other decoding, and we'll not deal with many techniques right now. We will do some notebooks. This lecture is to give a big picture of it, and so these are all some kinds of stuff that computational neuroscientists or recent people who are studying neuroscience are trying to do, and most of the works are related to using these techniques for getting better models of brain or understanding the brain. So in the recent conference we have seen enormous use of these things, and there is one more okay, so we can also use combination of encoding decoding models for this new technology which still didn't come up. It's still at the ideation stage, called neural co-processors. So you train a neural network which acts like a co-processor, and you have a brain injury, let's say. So bypassing that brain injury, there is like from some pre-region, let's say pre-motor area where you do motor planning, the activity is taken in, then it is converted to some stimulation parameters, some current parameters, which can then stimulate the other region when this connection is no more intact. Okay, so this can be used, and the error used to learn this is like motor error. How far are you from grabbing this? This whole error can be propagated here to make the network learn online. Or you can use, if you know the mapping between the planning versus motor cortex activity, you can use that as a loss function to learn this network. So this is another area, another advanced area where you can use both encoding and decoding models together to learn this. So there are other so in the conference we went, they mostly use this latent variable models and latent neural dynamics. So that gives more mechanistic insights into how meaningful are the neurons communicating to perform certain cognitive function or motor function. So we will get into that in the next class, but but it follows on these lines, on encoding decoding models, and then going to hidden variables, etc. So yeah, I think that's all for today. So yeah. So any comments, any doubts here? Okay, cool. So let's discuss the remaining part on Friday.